So here's a big surprise. We're going to talk about DNA a lot. And right now we're going to talk about making copies, both of the DNA and then of the cells that the DNA is in. Remember that DNA is a twisted ladder. What's called a double helix, where the rungs of the ladder are A's and T's and C's and G's. The connections are actually hydrogen bonds, so it's not a straight across bar, but it's okay to represent it that way. That's a little bit closer to how it works. It does give you an idea of the sizes. If nanometers makes any sense to you, it's a teeny, teeny size. Notice that there's the two ends are 5 primes and 3 primes. I don't want to get into why, but one strand is going in one direction and the opposite strand is going in the other direction. And that's how they get built. They get built in opposite directions. This is called a semi-conservative system. The conservative part is that the details are conserved, they're held onto. When you make a copy, your two copies are duplicates of the original. But the fact that it's, when you make a copy, you pull them apart and make a copy of what used to be on the other side, it's not a straight up match. It's not an A to A and T to T and C to C and G to G. It's semi-conservative. It is determined by what's on one side, but what's on the other side is different. And this is how DNA gets replicated. I'll talk about this, I'll write some more about this in a second. But it gets pulled apart. Okay, sorry, I just wanted to see where it was in my notes. There's a big old molecule sitting here, DNA polymerase, which will come up later when we talk about technology involving making copies of DNA from crime scenes and old bones and all sorts of things. But a DNA polymerase both separates out the strands and builds a new strand in one direction. So, and I'm just picking this at random. So it can build a new strand as it goes along. So it opens and builds a new strand, opens and builds a new strand. Well, it turns out that that strand is the one that builds backwards. So there's a secondary polymerase that has to build that way. So it's got to open a ways and then it builds some. And it's got to open a ways and then it builds some. It's got to open a ways and it builds some. And those bits and pieces that it builds from are called Okazaki fragments, from a Japanese researcher who figured this all out. So I'm going to write some stuff down, including some of the other enzymes that are kind of critical here. So this is DNA replication. As with anything, this is introductory stuff. It's more complicated than what you've dealt with in the past, but it's still not as complicated as it really is. In order for this to work, the twisted ladder needs to be untwisted quite a bit. And topoisomerase, it does the major unwinding. It's not completely ladderish by the time the other ones get to it, but it has started it, and then helicase comes along. ASE ending is going to tell you that it's an enzyme. And this separates it the rest of the way and starts to pull the strands apart. Primase, you need a little bit of RNA on there in order for the polymerase to attach properly, and the little bit of RNA is attached by the primase. And then you've got DNA polymerase. Which builds a second strand smoothly in one direction and in bits and pieces attached to one another in the other direction. But those bits and pieces are called Okazaki fragments. Relative to the DNA, polymerase is a big molecule. And so it's going along and going along and going along and going along. It's working its way down the DNA. DNA is being opened up. The two strands are being made behind it. It's going along, going along, going along. It hits the end of the molecule. Now, what it does not do is just go zoop right off the end and make copies of the strands that used to be there. What does happen is it gets to the end of the molecule, can't go any further. There's no primase out there. There's no RNA to give it a little bit of lead time. And so let's go. Which means that for this last bit of DNA, it's been separated, but the second side has not been made. It's literally a split end. And at the end of a chromosome, there's a cap. For reasons that we'll talk about a little bit later, but basically the short and sloppy way is so that your repair system doesn't look at the end of a chromosome and think it's a broken piece of DNA, a broken end off of a break. So the telomeres are pretty important, and now part of your telomere has frayed. You have another enzyme, telomerase. I forgot I already wrote it down. Telomerase comes in and finishes the job, finishes the cap, and all is well with the world. In cells that are in populations that divide all the time, telomerase is very active. In stem cells they sit in tissues of all different types, and they're kind of like amoeboid cells in cnidaria and sponges. They're there waiting to replace damaged cells, except they're a little bit more specialized. They tend to be there for different tissues, different types of stem cells. Here's a weird thing. In a lot of animals, as they get older, including us, the gene for this, epigenetically, gets less and less, it's a simple word, you get old, you lose simple words, activatable. You can't activate it. You can't make this stuff. And so you have the stem cell population, though the message comes in, oh, we need more cells, we need to replace some damage here. And the cells, first off, go, well, let's see if we can do this. And they check the robustness, the health of the DNA. They check some other things in the cells. And if they go down the checklist and they don't check everything, they go, sorry, we're not dividing. Try another cell. And in somebody my age, a lot of those stem cell populations, the telomeres have degraded. From division after division, they stopped making telomerase a while ago. And so now the DNA polymerase gets down to the frayed end and lets go there. And now you've got a longer piece that hasn't been doubled. And it hits a point where the cell goes, no, our DNA is too screwed up, we're not going to make more. And your ability to repair damage gets very compromised because you can't make more cells. This is one of the first enzymes that tends to get activated. We'll talk about this in the cancer chapter. But one of the things that cancers do from being really screwed up is they epigenetically do all sorts of weird things. They will activate genes that the cells wouldn't normally use that they came from. And telomerase, for them, is a great thing to activate. So they tend to activate telomerase, which makes them able to divide and divide and divide and divide and not have to run into this one level of this checking system. They have to actually exceed a whole bunch of other checks as well, but this is something that helps keep a cancer population of cells going, activation of telomerase. So we have two things that happen to people that we would like not to happen to people. The damages of aging and cancer, both of them associated with telomerase. The aging researchers are trying to find ways to get this stuff to turn back on so that you have a population of repair cells for damage. So your damage doesn't accumulate and age you to the point where eventually maybe you die. The cancer people would like the cancer cells not to be able to activate this, so they want to be able to counteract this. And if you're young and growing, you need this stuff active all over the place because you're making all sorts of new cells. But as you get older, except for things like your skin, as I said, populations where the cells are always dividing, this is always active. But in other populations where really the only divisions happen for damage repair, and even there's populations that never do that, you need that reactivated. And so the cancer people are saying, well, you aging researchers are gonna give cancer a leg up. They're not gonna have to activate telomerase if you're activating it yourself, and you're gonna see more cancer as a side effect. And the aging people are saying, well, if you turn it off, we're gonna get a lot more aging effects than we would otherwise. So both of them see the other side as potentially being damaging to their side. And don't mistake, if you're looking at places with a lot of money, a lot of research areas, cancer and aging. A lot of researchers getting older, they'd like not to be aging and dying. They're researching that. Of course, cancer is a huge funding pit for things. Just another picture of the replication. Just gives me a chance to say, when you get done, you're making a copy of a chromosome. Every new chromosome is half new and half old. Is that you use one side that was already there and you've made a new side on the other side. So you're not making, when you wind up with two chromosomes, you don't have an old chromosome and a new chromosome. Both chromosomes are half and half. Now here's something I ran into a few years ago. In the United States, as long as these two things are connected at the centromere, we call them chromatids. They're full chromosomes, but they're still stuck together. In Europe, once they're duplicated, they call them chromosomes. They don't differentiate the connected versus the, probably they say connected chromosomes. But these are chromatids until they do that, and then they're chromosomes. And the next part of the chapter is going to talk about cell division and how this all happens. And I'm going to stop here because that's a different section.